Good morning. My presentation today is entitled Hormesis, Biological Foundations, Medical and Public Health Implications. Now, the hormesis concept, at least from my perspective, addresses a broad framework of biological responses, but it focuses on low doses of numerous stresses. Very common ones, things such as exercise, intermittent fasting, hypoxia, heat, cold, ionizing, non-ionizing radiation, electricity, toxins, chemicals, drugs. All these entities, they can stimulate a wide range of adaptive responses. These induced adaptive responses, they have the potential to profoundly affect the success of public health practices and medical interventions, things such as surgeries, very common, chemo, radiation therapies for numerous disorders, for heart attacks, for stroke, for shock, brain traumatic damage, organ transplant. And we'll be trying to see how this fits into a dose-response framework. These stressors that can induce adaptive responses also offer ways to enhance healthy aging, to improve human performance, and to prevent damage to tissues later exposed to a wide range and levels of highly injurious stressors. At the center of this adaptive response concept, in my opinion, is the phenomenon of hormesis. Now, hormesis is a biphasic dose response that mediates processes by which human adaptation and performance may be improved. So what is hormesis? And how can it be used to achieve these public health and medical advances? And when and how should hormesis be taught to undergraduates and medical students? My definition of hormesis is that it's a dose-response phenomenon characterized by a low-dose stimulation and a high-dose inhibition. It's a non-monotonic, biphasic dose-response relationship with specific dose-response features, and that's really a very important component to it. It's not all biphasic dose responses, it really has a very specific quantitative features. I'm going to show you here kind of a standard dose-response models that are widely used within the biomedical community and the environmental regulatory community. Here we have a threshold model, which is seen within the screen. And as you follow the threshold model back down, you can see that there becomes a dose below which there is no biological effect or a real deviation from a control value. All you would really see below that threshold is background variability. You see a linear dose response model also represented here. There is an effect across the entire range of doses, and it gets proportionally less as the dose decreases. Now, in contrast to these two models, and these are the most dominant models that we have within the biological and medical sciences, we find now the hormetic model. And the hormetic model is like the other two in the high dose range, but at low doses you can see that there is a J shape that is introduced. And what that actually is meaning is that the effects are actually below those of the control value. So if you were looking at something such as disease incidence like heart disease or cancer incidence or mutation frequency, you'd be seeing at low doses there would be actually a reduction below the control value. Now, the hormetic dose response model is also represented by an inverted U, and we'll see this later. And it really depends upon the endpoint that you're measuring, whether you would represent it with a J shape or an inverted U. For example, many cases of inverted U's would be, if you were looking at plant growth or longevity, these would be typically represented by inverted U's, a low dose stimulation, high dose inhibition. Now, based upon studies over the past nearly 30 years by myself and others, we have created approximately 5 different databases on hormetic dose responses from the toxicological and pharmacological literature from various types of governmental databases covering a broad spectrum of endpoints and biological models. And numbering really in terms of dose responses in the 75,000 to maybe 100,000 fold range of hormetic dose responses. And what I've learned from an evaluation of this broad spectrum of findings is that generally speaking, the quantitative features of hormetic dose responses are very similar with respect to the amplitude of the response and the range of the stimulatory response. And this to me was actually pretty surprising, because when I first started exploring in some detail the hormetic dose response, I felt that there wouldn't be any restrictions on the amplitude. And I really never considered the width within the thinking. I was mostly focused on the amplitude. And what I found was, in fact, that the quantitative features are really remarkably similar from all the way from bacteria out to humans and everything in between, pretty much independent of biological, as I said, model, endpoint, the mechanism, things of this nature. It's pretty remarkable, and we'll try to address why that is the case as we go forth into this talk. It's also how it's produced. Hormetic dose responses can be directly induced, or they can be the result of compensatory processes following an initial disruption in homeostasis. However, regardless of the means of induction, the quantitative features are quite similar. Now, in the course of studying hormesis, there is a lot of debate over whether these low-dose stimulation responses, the hormetic response, are beneficial or harmful. And in my opinion, hormesis is really simply a dose-response relationship. Whether it's harmful or not should not be part of the definition of hormesis. This assessment needs to be made, but it should be reserved for a subsequent evaluation of the biological and ecological context of the response. And we'll look at what I'm talking about in really the next slide. This is a schematic representation of data that we found in an evaluation of several thousand anti-tumor drugs in a US National Cancer Institute database. And we were interested in not the capacity of the anti-tumor drugs to actually cause decrease in growth or proliferation of the tumor cells. We're actually interested in whether at low doses these anti-tumor drugs could stimulate the growth of tumor cells. And almost invariably, this was the case. And if you were to try to graph these, they would essentially almost always show this inverted U-shaped dose response, the hormetic dose response. And so if you just drew back the dose, made it more dilute, these relatively toxic anti-tumor drugs would become stimulatory in the low dose zone. Now, if you take a look at a very large other set of studies dealing with memory enhancing drugs, and these coming from studies in rodent models. Here we have a situation where at low doses there is a stimulation or an enhancement of the memory response, but at higher doses, essentially this desired response becomes inhibited. So if we compare the past slide with this one, we find that at low doses, the anti-tumor drugs are actually inducing an effect that is undesirable to the patient. Whereas in this particular case, the low dose enhancing memory would be a desirable effect. So a hormetic effect can be both, or either be beneficial or harmful depending upon the context and how you're interpreting it. We study hormetic dose responses in multiple ways, experimentally and evaluating dose response patterns, doing statistical evaluations. There's a great need, as we know, by having to replicate findings, and we'll get into reasons for enhanced replication needs later on. Also, there is a profound need to have mechanistic documentation, and the idea of simulation studies is very important when you have large databases like that NCI database that I mentioned, because when you have normal background variability in studies, background variability could be anywhere from less than 5% to 20% or more. And just because you have background variability, even when you have no treatment effect, there will be the possibility that a hormetic effect could occur just by chance alone. And so one has to be very aware of the possibility of false positives. And when I use or do studies or evaluations of large data sets, they're usually done in close connection with various biostatisticians who are typically very much involved with simulation studies, providing oversight and check into our interpretations, making sure that we do not falsely conclude that there is a hormetic dose response when in fact it could be explained by this background variability. And so essentially these are various types of methodological approaches that we use in the assessment of hormesis. In general, what I found as a result of looking at all these hormetic databases with many thousands of examples is that this is a very general finding that hormesis occurs widespread within the biological literature. You find it within plants and microbes and vertebrates, humans. You find it within vitro studies, in vivo studies. It's independent of endpoint, it's independent of the inducing agent, and it's also independent of the level of biological organization. You see it at the cell level, the organ level, and the individual level. And something that's not on the screen there, but we showed later, and that we eventually were able to show that it's also independent of mechanism. It really doesn't make any difference which particular receptor that is involved, or the cell signaling pathway, or whether it's receptor mediated or not. The quantitative features of the hormetic dose response are similar. It's a really interesting situation. How in the course of evolution would in fact the strategy be that this dose response relationship would be so highly conserved and play out within all these different types of experimental parameters is really an underappreciated, overlooked, but really fundamental observation, and it needs to be fit within an evolutionary perspective. Now, the hormetic dose response, as I've suggested or alluded to, is actually pretty modest. It's only in the max, it's only in the 30 to 60% greater than control. Usually you don't see it occurring in a sense more than twice the control group value. Now, when you have the maximum response only being at 30 to 60% greater than the control, it's very easy that you would have a response just by background variation that could go into that range. And so that's why it's very important when you're studying hormetic dose responses that you actually have a relatively large number of doses, that you have an adequate number of doses in the low dose zone, and then you want to see this dose response pattern, and you want to see it reproduced. That's why there's such an emphasis on reproducibility of the findings and a need for adequate statistical power. When I first got involved with my initial observations with hormesis in an experimental context, my advisor at the time, because I was a student, my advisor at the time essentially required me to replicate those findings approximately 10 to 12 times in progressively larger experiments with ever more subjects in the low dose zone and doses in the low dose zone. Ultimately, it was the consistent replication of the findings under stronger and stronger study designs that led this person to ultimately have confidence in the findings. And this is one of the difficulties with the hormetic dose response, and that is that you have much greater need for replication that requires more time, more resources, but ultimately this is what has to be done if you want to have confidence in your findings. The width of the stimulatory zone has been much more variable. However, most of the findings are within about 20-fold of the threshold, or what we call the NOEL, the no observed effect level, or no observed adverse effect level. In some cases, about 1 or 2% of the cases, we find that the stimulatory range can be very wide, can go out to well over 1,000-fold. And so these, whether it's very narrow or whether it's very broad, it can have a very significant public health medical implications, and we'll talk about that a little bit later. This is a standard dose response that represents the inverted U version of the hormetic dose response. And you can see in this graphic framework, the dotted line is really the control group, and everything is normalized to that for this particular comparison. And you can see that as you go a little bit out on the on that control line on the dose framework, you begin to see that increase in response that peaks around 30 to 60% above the control value and then drops down below the control. You can see the very narrow stimulatory range. And so this is it's really a small signal, relatively speaking, and that's why I think it's been very difficult for people to come to understand hormesis or to believe that it's real, because to actually study it, you need to have very strong study designs, and ideally, it's also better to have a control group that has a very small background variability, so that essentially you can have a nice signal to noise ratio. But these are different types of strategies that you would have going forth, in an experimental approach. In the course of public health evaluation, some people will ask, and have asked me, does hormesis occur in all segments of the human population, those who are normal and those who may be at increased risk? And so our databases have allowed us the opportunity to go back in and to explore the occurrence of hormesis within the framework of a high degree of inter-individual variability that would include both normal and high-risk groups. And indeed, what we've found is that the hormetic dose response occurs essentially in all people, or all subjects within the evaluation. Those that are more resistant to the condition, or you might say in the normal framework here, they will express that hormetic inverted U-shaped dose response over a at higher dose within the dose response spectrum. Whereas for the high risk group, it'll be shifted to the left, and that's what I'm trying to represent in this particular slide. This has very significant implications in the environmental side, because if one was trying to optimize a response within a human population, you can see that the peak of the hormetic stimulation in the normal group is actually at a dose that is actually having a very inhibitory or potentially harmful effect in the high risk group. And if you wanted to normalize or optimize on the high risk group, it would be so low that it wouldn't benefit the normal group. And so hormesis actually provides much more challenging circumstances in public evaluation than the standard LNT or threshold dose response model. What I'm showing here are a representation of different types of receptor systems that display biphasic dose response relationships or hormetic dose response relationships. I won't go through all of them, but I have this long list just to provide you with an indication that hormetic dose responses affect essentially all systems, and every single important endpoint that goes on within a cell and within your body. In my opinion, there is evidence to support that it's actually mediated by hormetic dose responses. It shows its universality, its generality. Now, hormetic mechanisms have become very much in focus within the research community. Back in the 1990s, there was a lot of work demonstrating hormesis and its reproducibility, but the underlying mechanistic foundations were not very well established. However, since the early 2000s, and certainly very evident today, 20 years later, we find that hormetic mechanisms are included in essentially just about every study that's published concerning the dose-response relationships relating to hormesis. And in 2014 or 15, we published a paper that had 400 different hormetic dose-response relationships, identified the mechanism down to the level of receptor and cell signaling pathway. And in the course of doing this evaluation, we found that it really didn't make any difference what the mechanism was. The quantitative features of the dose response are really basically the same. And so whether it's receptor, cell signaling pathway, their interaction, or others, it all basically conforms to these quantitative features. And we'll try to address what is going on and why is this the case. So regardless of the mechanism, the quantitative features really stay the same. Now, hormetic dose responses, when you look for them, are very broadly found within, by endpoint within the biological literature. For example, these are some of the endpoints. Any endpoint that is integrative and significant, you'll see a hormetic dose response commonly found. Things such as cell proliferation, fecundity, tissue repair, behavior, learning, response to disease, injury resistance, the area of preconditioning, postconditioning, DNA damage, tumor incidence, aging, longevity. These very significant endpoints are really just flush with hormetic dose responses within the scientific literature. This is a picture trying to integrate some of the information that I've just mentioned, showing the inverted U shape of dose response, and within that, trying to highlight to you, that underlying this response is a whole series of adaptive mechanisms that allows these integrated responses to actually become manifest. Now, I'm going to share with you some examples of hormetic dose responses. And these were selected not because they're the best, so to speak, but they were selected just to show you the range and the generality of the hormetic dose responses, so that you can see that it's not truncated in any one particular area, but occurs in a broad range of biological models and a broad range of endpoints. And the basic dose response strategy, cellular strategy, organismal strategy, ultimately plays out with the same quantitative features of the dose response. Here you have a case of the effects of ethanol on social activity, social behavior in adolescent young adult rats. So they're at age day 30. And you can see that in this dose response relationship, the dashed line represents the control group line. So everything is compared to the control group. And you can see as the dose goes up, you can see that the social activity and playfulness of these rats, because I've seen these studies, I've seen the rats, and they become very suppressed at high doses. But as the dose gets reduced, you can see that there's a flip to the other side of the control value, and their activity becomes significantly increased. And it follows this inverted U-shaped dose response. It conforms very nicely to the quantitative features of the hormetic dose response. This is an example of a fruit fly longevity study using the highly toxic chemical methanol. And here you can see, using male and female fruit flies, that at high doses, the methanol has a very significant dose-dependent effect, reducing the fruit fly longevity. But as you lower that dose, for both male and female, the response flips to the other side again, and you have an enhancement in longevity of by about 20% for males and females. These findings, as the others, are really highly reproducible and striking within the world literature. In this particular case, the third one, we have an example of alixin, which is a key ingredient in garlic, and it's being assessed here on the survival of primary cultured hippocampal neurons in the 18-day embryonic embryo of or fetus of the Wistar rat. And you can see at high doses, the alixin is harmful for the neuronal survival. But drawing the dose back to a more dilute form, it becomes stimulatory and displays this hormetic dose response quite well, again, with a maximum in the 30 to 50% zone. So it's very, very consistent with these other examples. In this particular case now, you have the effects of sodium arsenate on lymphocyte stimulation in bovine lymphocytes. Here we have 10 or 12 different concentrations which are being studied, and I'd ask you to focus on the higher doses, and you can see a nice dose-dependent decrease in the lymphocyte stimulation, but draw that dose back to the more dilute, and you see it flips over again to the other side, and you have this low-dose stimulation, high-dose inhibition. You have it maxing out at around the 30 to 50% zone, and if you never did the low doses, you would never see that stimulation, and you just kind of infer back to, gee, there would be no biological activity below that threshold, but we see that you actually do have biological activity below that threshold. In this particular case, we're now looking at methylmercury, and methylmercury is one of the most toxic agents that regulatory agencies focus in on. A lot of concern with human health for this, neurological concerns. And here we're looking at a neurological cell line, a D407 cell line. But what we see here is, not unexpectedly, at high doses, this is highly toxic, adversely affecting viability of that cell line. However, at low doses, what you find is that the methylmercury, this highly toxic neurotoxic agent, is actually enhancing the viability of these cells. It conforms very well to a hormetic dose-response relationship. And I like this particular study design because it actually had 4 doses, 4 concentrations in the hormetic zone, all of them being statistically significant, but this is a highly reproducible assay, highly reproducible endpoint here. And that begins to give you more confidence that this biological response is not due to chance, but actually due to the real reproducible effect. Here we're looking at the effects of nicotine on the proliferation of human osteoblast-like cells. Now we know that heavy smoking is a cause of osteoporosis, but it's interesting when you take a look, and people have related it in part to high levels of exposure to nicotine. In this particular case, what we're seeing is that nicotine actually also, for this osteoblast endpoint, shows a biphasic dose response. So at high doses, it would be preventing this proliferative response, something that could be perhaps suggestive of leading to an osteoporosis type response. But at low doses, it actually enhances this response. People have seen these kinds of data and identified nicotine as a potential agent that could, at low dose, could actually prevent the occurrence of osteoporosis. And in the world of making money, people have then tried to make nicotine look-alike drugs that they could market, to pharmaceutical companies. This particular slide has a lot of little bells and whistles on it, but it's really trying to show you, again, the effects, hormetic effects of sodium arsenite on cell proliferation in human embryo lung fibroblast. And you can see the biphasic dose response, but you see these little white spots along in the slide. And what they're trying to show you here is a mechanistic link up to this slide. So for example, researchers attempt to identify receptors or pathways that may be involved in mediating this stimulation or inhibition. And what I've represented to you here is when you use certain types of inhibitors, you can essentially reverse this stimulation and bring the response right back to the control group. And that will tell you that, in fact, that receptor or that pathway is mediating the stimulation. You can see that it also works on the inhibition as well. And so this is a way that essentially most papers today that are published that are looking at hormesis and link up to mechanism, where they try to identify receptors or pathways and their interaction that allow one to explain this dose response. And this has further enhanced the acceptance and the credibility of the hormetic dose response, because it essentially is just fit into mainstream toxicology and pharmacology within this mechanistic framework. Here we're looking at the effects of morphine on a rodent seizure model. And in this particular case, this drug called PTZ is used to induce a certain type of seizure. And you can see here that the morphine is making it about 25% more difficult for this PTZ to induce a seizure. It's requiring that much more drug to induce a seizure. That makes morphine a potential anti-seizure drug candidate. When I looked at this dose response, I saw evidence for hormesis. 10 or 12 doses, a beautiful inverted U-shaped dose response, and this led me into the anti-seizure drug literature. And essentially, just about every single drug that I saw in preclinical testing showed this inverted U hormetic dose response. So in my opinion, essentially almost the entire field of anti-seizure drug pharmacology was being mediated by hormetic dose responses, and the field really never mentioned the term hormesis, yet it was the capacity of the drugs were always restricted to this 25 to 50%, 60% increase. It was all conforming to a hormetic dose response. You see the same thing with anxiolytic drugs. This is an example here using DHEA, but we're using essentially a mouse model, and we're trying to evaluate whether the drug has the capacity to reduce anxiety in the drugs. Now, rodent models, rodents in general, they are reluctant to go into lighted areas, open areas. To the extent that you can make them go into those areas or encourage them to go into those areas, it's interpreted as reducing their anxiety. And so here what we see is that this drug is able to increase the frequency with which the animal will go into these lighted areas. And it follows an inverted U-shaped dose response. So when it reaches the peak, as you can see, this is interpreted as having significantly reduced the animal's anxiety. And you can see that at the higher doses, the drug actually increases the anxiety. And so it follows a beautiful hormetic type of dose response relationship. And I've looked at probably about 175 to 200 anxiolytic drugs with respect to their preclinical findings. And essentially every single one follows a hormetic dose response. So the whole area of anxiolytic drugs, anti-anxiety drugs are all based upon the hormetic model. Here we're looking at the chemical resveratrol, which has a lot of publicity in the general public today, found in certain quantities in red wine and so forth. And you can see that we're looking at cardiovascular endpoints at high doses, or concentration doses here. It is actually increasing damage, but at low dose, you can see this J-shaped curve, and it's reducing the occurrence of myocardial infarct damage and cardiomyocyte apoptosis. I want to now move over to the area of preconditioning hormesis. And preconditioning is when you would give a low dose of a drug a day or two before, or a stressor agent, before a high dose that may come subsequently. And the high dose, the low dose, if you have the low dose, it ultimately upregulates adaptive mechanisms that can protect you from subsequent damage from this more massive exposure. Now, in getting into this, the term preconditioning was really created in 1986 by researchers at Duke University who used a very mild, repetitive, I'm going to call it a type of hypoxic sort of a stress, and then ultimately exposed dogs to a massive heart attack, and found that this blocking off of the oxygen to the heart the day or two before resulted in profound protection. And they called this a preconditioning effect. The findings were replicated. They were extended to many rodent models and rabbits and sheep and so forth, and found to be very general. And it was also ultimately found not only that you could induce it by a direct experimental surgical means, but you could actually induce it by in many other ways. Chemically, you could do it physically. People have used this remotely using a blood pressure cuff on your arm, pumping it up to 200 millimeters of mercury, holding it for 3 minutes, repeating it several times, and you would get the same benefit as if you had done surgical manipulation. It's a very significant advance. People in other fields have also found this using low doses of radiation, given before more. Massive dose upregulates adaptive mechanisms and is very protective. It was actually first reported in the literature back in 1928 using plants, and low doses of radiation could protect the plants against subsequent exposures. And this is a very significant phenomenon. And what it's really doing is it's upregulating adaptive mechanisms and resulting in essentially acquired resilience, which I believe all conforms to a hormetic dose-response relationship. This is an example of, on the slide here, using hydrogen peroxide pre-treatment, and it protects the PC12 cells against an oxygen glucose deprivation stress. And what we see here in the slide in a J-shaped form is actual protection of the cells by about 50%, depending upon the level of what the acute dose was. The stimulation in the inverted U is actually a measure of BCL2 expression, which is one of the upregulated adaptive mechanisms. Over here, we see the same kind of situation, again using hydrogen peroxide, but we're using also now hydrogen peroxide is low dose of hydrogen peroxide protecting against high doses of hydrogen peroxide. And you can see when you pre-dose it 24 hours before the big dose, the damage essentially goes down by about 50%. But if you increase the dose level of the preconditioning doses, you can lose your protective effect. So in my opinion, and we found hundreds of examples for this, that the preconditioning really is a manifestation of hormesis. And if you have enough preconditioning doses, they follow this hormetic dose response. This is another example using nitrite and protecting against heart attack damage. And this is an example with camptothecin. And what it does is it's a direct stimulatory response on the cells and these PC12 cells in question. But you'll see the same hormetic response in the lower part of this slide if you have pre-stressed it. And so essentially hormesis is occurring with a direct stimulation or after within the context of a preconditioning protocol. Now, key observations here is that we have numerous pre and post conditioning studies display hormetic dose responses with mechanistic explanations. Thus, pre and post conditioning are manifestations of hormesis. I want to briefly show you hormesis and cancer. Here we have gamma rays and mouse lung adenomas. We find that as within this framework that actually low doses of radiation decrease the occurrence of the lung adenomas in males and females. Each of these boxes, the squares and triangles, they have about 500 to 750 mice per box, per triangle. So it's a very substantial study. And it shows that for this cancer endpoint, that it's best explained by a hormetic dose response. This is a study in Japan dealing with low dose radiation at about 60 to 70 times above background throughout their whole life. And one can see at 92 weeks that we have the exposed groups have 70% alive, whereas the control groups have only 40% alive. You'd expect it to have been totally reversed. But I want to show you here on the slide a representative animal from both of these groups. You can see the control group looks pretty much near death. It's lost its fur. It looks unhealthy. It's something that's you know, is really quite unattractive and quite unhealthy. However, the group getting the low dose radiation is shiny fur, full fur, plump, and essentially looks much healthier than that control group. And really, the only difference between them is the low dose of radiation. So it kind of told me two things. One is that if you live the so-called good life as the control group, it's not so good at all. And then it also is telling me that, in fact, that we can do something to enhance our resilience to the effects of aging and perhaps underlying disease conditions like this diabetic predisposition. And this is something, you know, that we as people interested in public health and medicine need to explore, in the advantages of a hormetic framework. This over here is showing you the effects of gamma rays on the lifespan in the female cricket. And again, it's showing high doses are harmful, but low doses are beneficial. This is showing the same sort of situation with gamma rays and mutation. High doses are actually inducing the mutation, but low doses are actually decreasing it. It's in total contrast to what regulatory agencies would in fact want us to believe. Here's an example of DDT, which is really a highly maligned chemical. At high doses, it causes liver foci and is a carcinogen, but Japanese researchers have shown that at low doses, it actually decreases the risk of liver cancer. Very reproducible study, very mechanistically oriented. In a mega mouse study in the United States with 24,000 animals, looking at the carcinogenicity of AAF, we find that at high doses, yes, it is a bladder carcinogen, but at low doses, in fact, you see this hormetic dose response relationship. So what is hormesis actually indicating? It's showing you that the low dose stimulation is different than high dose inhibition and toxicity. It's showing you that this low dose stimulation is a measure of biological performance, not toxicity. And it's determining how much a system can respond. What is hormesis actually indicating? It's really the first quantitative estimate, in my opinion, of biological plasticity, and that the hormesis stimulatory response is really constrained by the limits of plasticity. Hormesis has many applications, but in the drug area for anxiolytic drug, anti-seizure drugs, memory drugs, osteoporosis, wound healing, really, and many others. It's just a very general and evident and important development. It can be useful for medical procedures involved with for organ transplant optimization through preconditioning approaches, where you could stress systems to upregulate adaptive responses and to better resist the stresses when you have the major surgical interventions. Here we have other factors such as diet, intermittent fasting are examples of preconditioning modes such as exercise. They all conform to hormetic dose responses. In the case of regulatory aspects, the nadir of the hormetic dose response actually conforms very closely to a 1 in 10,000 risk using the LNT model, and would actually correspond nicely to using a threshold model with animals that had the traditional 2 safety factors. They all line up on this hormetic nadir, and I believe this is the confluence of 3 different models. And I think that this may be the regulatory sweet spot, which optimizes public health, and it could be driven by a hormetic perspective. So my conclusions are for you today that hormesis is a general and central biological concept. It affects all disciplines utilizing the dose response concept. It represents a general adaptive strategy through which biological performance is enhanced and mediated. It provides both the theoretical basis and the molecular foundations to create biological shields that can protect against chemical, radiological, and microbiological threats. It has implications for numerous and diverse medical interventions, as well as affecting the health and performance of military personnel, athletes, the general public, including patients and accident victims, and others. Failure to consider the hormetic dose response within hazard assessment and risk assessment, in my opinion, has been a very serious failing of modern risk assessment. And risk assessment needs to consider the entire dose response continuum in order to serve the public health. Hormesis can and should play a significant role in drug discovery, development, and evaluation, and it should become fully integrated into educational programs for medical school students and continuing educational programs. And I'd like to thank you very much.